Hey everyone, so we're moving on from cell signaling and we're going to start talking about the cell cycle. And cell signaling is going to come back around in a little while, but we're going to talk about how it relates to the cell cycle. But first, let's talk about some general cell division, cell cycle stuff. So first off, I want you to think about what might be happening here. So in this picture, there you can see an octopus arm, and that octopus arm was cut off. And then in this picture, you can see kind of where that octopus arm was cut off. In the subsequent pictures, you can see that it's starting to regenerate that arm. And then you get down here at the end, you get the almost completely regenerated arm. So obviously this regenerated arm here is going to be new tissue. So I want you to just pause the video for a minute, talk to the person next to you, and think about, okay, where did all of that new tissue come from? All right, and we're back. So I hope that you realized that the new tissue is made up of new cells, and those new cells had to come from existing cells replicating and dividing. The cell cycle deals with how that replication and division is happening. So we're going to back up for a few minutes and then we're going to come back to cell cycle, more specific aspects of the cell cycle. When we talk about cell division, one of the ways that we have to divide is to reproduce. And we have to divide cells to produce reproductive cells, or in the case of asexual individuals are just going to divide themselves by binary fission, potentially. So what happens with binary fission is the organism, usually like a bacteria, will grow, and then it will replicate everything inside of it, and then it will actually just split off. So it replicates its DNA, it grows in size, it separates into two cells, and then those two cells are genetic clones of each other. Asexual reproduction is reproduction that happens without a partner. So you can see this happening in single-celled organisms, but you can also see it happening in multicellular organisms. And I'll show you a video when we get back about how this can happen in sea stars. Sexual reproduction is different. Sexual reproduction is reproduction that happens with a partner, and you get the fusion of two specialized cells or gametes. And those gametes are formed by meiosis, which is what we're going to talk about later on when you guys get back. Right? But how this works is that organisms are 2N, meaning that they have two copies of each gene, and therefore each chromosome. So the 2N individual has to be able to produce N gametes, right? So you might have like a sperm that is N, and then an egg that is N. Right? So in the process of meiosis, which we'll talk more about later, you take a 2N cell and then you make N cells. So this is a diploid organism, diploid cell, and these are going to be haploid cells. Those haploid cells will then have to fuse together to create a zygote, which is going to be 2N. If this didn't happen, if we didn't actually have the genetic material in the gametes, if we had 2N cells fusing with 2N cells, then every generation we'd have a doubling of the genetic material, which could be really problematic. So in this case, the amount of genetic material stays consistent across generations. So this is what's occurring, again, in sexual reproduction. So when does cell reproduction or division actually happen? When do cells actually know when to divide? And when they are going to divide is going to depend on if it's a prokaryotic cell or if it's a eukaryotic cell. And we're going to talk about that in more detail in a minute, right? It has to do with environmental conditions, or it could be related to the needs of the entire organism. It could also involve growth factors, which are signals from potentially neighboring cells or other cells in the body. So growth factors in mammals will stimulate cell division and differentiation. And we're going to talk about some more specific growth factors, more specific cell signals that will lead to cell division later on. You can see in this picture here that the medium without growth factors did not induce cell division, and the medium with growth factors did induce cell division. When cells are getting ready to grow and divide, one of the things that they have to be able to duplicate and divide is their genetic material. And the division of the genetic material, the replication and the division of the genetic material, is a really important component of the cell cycle and cell division that we're going to talk about more specifically. In order to talk about how the genetic material divides, we need to also talk about how the genetic material is organized. So we've talked a little bit about DNA as a macromolecule, and you can see here's the sugar phosphate backbone, which is that blue material, and then here are the nitrogenous bases on the inside. And we talked a little bit about how the order of the nitrogenous bases and their pairing is a code, and that code gets read and it will be used to make proteins. These DNA molecules are extremely long, and you can imagine this as like a giant string, and if we just had our DNA organized as this ginormous string in the nucleus of our cells, or just within the cell if you're a prokaryote, it would be really messy, right? Like that's going to get tangled, it's going to be hard to read it, it's going to be hard to divide it, it would just not be ideal. So what we want to do instead is we want to take this very, very long DNA molecule and we want to compact it down into a chromosome. And we do that using histones. So histones are proteins that provide structural support for a chromosome. So the DNA will actually wrap around the protein histones. And then when the DNA wraps around the protein histones, you get these complexes right here called nucleosomes. So nucleosomes are this basic repeating subunit of chromatin, or DNA and its associated proteins, that are packaged in the nucleus. So those nucleosomes, these little units, will get condensed down into coils, so they'll start coiling around each other, and then that coil becomes condensed into the chromosome. So the chromosome is just basically a really condensed version of the DNA molecule using proteins. So some terms to be aware of: histones, which again are the proteins, then nucleosomes, right, which are the protein DNA complexes, and then chromosome, which is this condensed version of the DNA and its associated proteins. All right, we're going to have to add in a little bit more terminology in how we describe chromosomes. Let me push this off to the side. When we talked about being diploid organisms, we talked about how we are 2N, which means we have 2 copies of each gene, and therefore each chromosome. So you have a maternal copy of each chromosome, and you have a paternal copy of each chromosome. One from the egg and then one from the sperm. Now, what is a little bit tricky to understand about these chromosomes is that they are coding for the same genes. So what that means is this section of the paternal chromosome might code for the eye color gene, which means that the maternal chromosome, the same section of the maternal chromosome, is coding for the eye color gene. So that means you could have a different set of chromosomes. So these ones are much smaller over here because they're different chromosomes, right? Maybe these ones are over here and the other ones are over here. So this other set of chromosomes, maybe on this one you get the gene for hair type. So therefore, on the maternal chromosome, you would also have the gene for hair type. We'll get into genetics and how the combination of those 2 copies will make a difference, but just to clarify this a little bit, you could have gotten the brown-eyed version of that gene or allele from your maternal copy, and you could have gotten the blue-eyed version or allele from the paternal copy. So you can have different versions of the same gene, but the actual gene for eye color is located in the same spot. And then the combination of those 2 alleles or versions of the gene will determine what kind of trait you have. So I'm oversimplifying eye color a little bit, but in this sense, if you had a brown copy and you had a blue copy, then you would actually have green eyes because it's the combination of the 2. So this maternal and paternal copy that code for the same genes, these are called homologous pairs or homologous chromosomes. So these two are homologous chromosomes or homologous pairs. And these two are also homologous pairs. So each homologous pair has a maternal copy and a paternal copy. So hopefully the homologous pairs of chromosomes make sense, because now we're going to add a little bit of a additional piece to it. So when a cell grows and divides, it's going to replicate all of its genetic material, and then it will produce two daughter cells. So one cell is going to become two daughter cells. And those two daughter cells are each going to have a maternal copy and a paternal copy of the original homologous chromosomes. So these homologous chromosomes have to replicate. So what this will look like is the maternal copy is going to replicate, and then the paternal copy is going to replicate. These two are still maternal, and these two are still paternal. The two maternal copies are identical to each other, and the two paternal copies are identical to each other. So we would say that these two identical maternal copies are sister chromatids. And then we would say that these two identical paternal copies are sister chromatids. If we think about this in terms of the eye color gene, it's still there, right? So this eye color gene here is the same as this one here. And then this one here is the same as this one here. So the sister chromatids are identical to each other, the same allele for the same gene. The sister chromatids are also attached to each other here and then here, and where they're attached is called the centromere. So what is this actually going to look like when we think about division? And we will look at this in more detail. So if here's your starting cell, right, we're just going to look at this with one set of chromosomes, and you're starting off with a pair of homologous chromosomes like this. Right? Those homologous chromosomes, if that cell goes through division, so this cell is going to go through division, right? When that cell starts to go through division, it's going to replicate that homologous pair of chromosomes. So now we have two sets of sister chromatids, and those sister chromatids are going to be attached to each other at the centromere. When this cell goes through division, it's going to actually pull apart those sister chromatids, and you'll get the gathering of one of the sets of homologous chromosomes in each part of the cell. That cell will then divide into two cells, and each of those two cells is going to have one complete homologous chromosome pair. So you can see here you went from 2N to essentially 4N, right, because of the sister chromatids, and then you end up with two 2N cells, just like you had at the start. Our last term to know is the karyotype, which is this diagram right here. And the karyotype just looks at all of the different homologous pairs of chromosomes. So for humans, we have 23 pairs of chromosomes. One of the pairs is the sex chromosomes, which we will talk about later. But for each of these homologous pairs of chromosomes, you have a maternal and a paternal pair chromosome. Maternal, paternal, maternal, paternal, and so on and so on. So in a general sense, we want to talk a little bit about what has to happen in cell division. And first we need a cell division signal, then we need to replicate DNA, then we need to segregate the DNA, which we'll talk about in a second, and then we need cytokinesis. So the way these things happen can differ a little bit in prokaryotes versus eukaryotes. So what I'd like you to do now is maybe pause the video and make a big table in your notes like this. And then do prokaryotes and eukaryotes. So cell division signals are where one or more signals are going to be required to initiate cell division. So signals to initiate division. And these could come from either inside or outside the cell. In prokaryotes, they're going to be signaled to divide if environmental conditions and nutrient concentrations are adequate. With eukaryote, we could be stimulated for cell division to happen if environmental conditions are favorable. But we could also, this could be more related to internal conditions stimulating division. And that could be largely related to function and specialized cell types in some cases. Like if you need your liver cells to divide, you want maybe just your liver cells to divide. You don't want every cell in your body to divide when just that specialized area needs to divide. We also need DNA replication to occur so that the genetic material gets duplicated. And this is so that each of the two new cells are going to have a full complement of genetic material. If we start out as a 2N organism, so we have 2 copies of each gene, 2 copies of each chromosome, if we didn't actually replicate that genetic material, then we would end up having the genetic material and we would go to N and then half of N. So that could become really problematic. So we want to maintain the 2N diploid status. So in prokaryotes, which we talked about at the beginning of the year, these guys have one singular circular chromosome. So it looks like this, which means it's going to have one origin of replication because it's just going to have that one origin. It's going to replicate all the way around the circle and then end. Prokaryotes have more than one chromosome. So this is going to be more complex, and they're going to have multiple origins of replication. After the DNA or during the DNA replication, we need that replicated DNA to be distributed evenly between the 2 daughter cells. So DNA segregation is going to be even distribution of replicated DNA. In prokaryotes, segregation and replication can co-occur, so happen at the same time. So as the circular chromosome is being replicated, it can also be segregating to opposite sides of the cell to get ready for division. In eukaryotes, the segregation, since there's multiple chromosomes, can be more complex. So you need to end up having one copy, as we looked at in the previous part, one copy each chromosome from the parental cell must end up in each new cell. Lastly, of course, it's not just important that the DNA gets replicated and divided. The cytoplasm and everything else has to also get divided to form 2 new cells, and each of those new cells needs to be surrounded by a cell membrane and or a cell wall. So in the cytokinesis piece, cytoplasm dividing into 2 new cells and cell membrane formed and potentially cell wall. So in prokaryotes, you get this pinching off of the cell membrane, and you get the peptidoglycan cell wall formed. In eukaryotes, cytokinesis is going to happen in the M phase of the cell cycle. I know we haven't talked about that yet, so we'll just talk about that. Just know that we're going to talk about that in just a minute. So this is the cell cycle that I keep alluding to, and the cell cycle is made up of 4 stages: G1, S, G2, and then M. And we're going to talk a little bit about what's happening at each of these phases of the cell cycle. So if you can remember that G means growth, that could be helpful for understanding what's happening in some of these stages. So in the G1 phase, which is right here, G1 phase, the cell is growing larger. Because you can imagine if the cell didn't grow larger before it divided, then we would have subsequently smaller and smaller and smaller cells for every division, and that would not be ideal. So in the G1 phase, it's going to grow larger to prep to divide, and it's going to copy its organelles. Right? Because we also don't want to have the number of organelles in our cells, because that would also be problematic. If you can remember that S stands for synthesis, right here, the S phase is where the DNA gets copied. Right, so that's where DNA replication is occurring, or DNA is being synthesized. The G2 phase is where the cell is going to grow more, right, and it's kind of reorganize and prep for division. The M phase is where mitosis is going to occur, and we're going to talk more detail about mitosis later on, but mitosis is the part of the cell cycle where the division and segregation of the genetic material is going to happen, and we'll talk about the order of that. But mitosis is the division and segregation of that replicated genetic material. And then also happening in the M phase is cytokinesis, which is this physical division of the 2 daughter cells. We're going to go into this next part a little bit more detail later on, but within the cell cycle there are 3 checkpoints here, here, and here. And at those checkpoints there are genes that produce proteins that are going to be making sure that the cell cycle is happening correctly. So making sure that the cell is growing enough, making sure that the DNA is being replicated, making sure that there's no DNA damage, making sure the cell is big enough. All of those checkpoints make sure that we're not going to be producing damaged or cells that are going to have problems with their genetic material. You can imagine that problems with the genetic material could result in things like cancer, and we're going to get into that later on. This first checkpoint is the G1 checkpoint, and this is going to this is actually the main decision point for the cell. It's going to decide whether or not to divide, and cell division is going to be irreversible. It's going to be committed to cell division if it passes this checkpoint. It's looking at whether or not the cell is big enough. It's looking at whether or not there's enough nutrients and energy supported to divide. It's looking at whether or not it's getting the correct positive cues or growth factors from neighbors in order to divide. And it's also going to look at DNA integrity. Is the DNA damaged before it's going through division? The next cell cycle checkpoint is here, and that's the G2 cell cycle checkpoint. So you can see a lot has happened between these 2 checkpoints, right? You've gone through DNA replication, and then the cell has grown some more. So at this point, it's looking at, again, at DNA integrity. Is the DNA damaged? Did the DNA copy completely in that S phase? So if problems are detected, sometimes it will pause to repair the damage. If the damage is irreversible, it will undergo programmed cell death, which is called apoptosis. The last cell cycle checkpoint is this one right here, and this is the spindle checkpoint, and this is happening in the M phase, so during mitosis. This is looking at whether or not those sister chromatids are correctly attached to microtubules, because those microtubules are going to be pulling the sister chromatids apart so that they segregate into different cells. So needs to make sure that those sister chromatids are going to be pulled apart correctly so that they segregate correctly into the 2 daughter cells. All right, that's it for now.